One of the most striking features of the living world is the great disparity observed amongst living organisms. Even closely related organisms within the same species still differ in size, shape, and form. Have you ever wondered why parents and their children, or even more striking, why siblings from the same parents, though sharing certain common features, do not resemble each other exactly? Well, the answer lies in genetics. Genetics is a field of molecular biology that deals with the study of variation and inheritance. In this chapter, we shall understand why organisms differ from each other and at the same time show similarities within the same species. Remember, only organisms within the same species can freely interbreed to produce viable offsprings. These offsprings resemble their parents in form. Why? Because they inherit these features from their parents. However, differences are seen between parents and their offsprings. First, obviously because the mother and the father are not alike. But then, why would siblings from the same parents be different? Maybe in skin color, height, hair color, eye color, and so on. It is because of a variety of hereditary factors that children acquire from their parents during fertilization. The children do not inherit the exact same factors from their parents, and this results in the siblings being different in some ways. Another thing that we shall understand is that not all the factors inherited are expressed physically. For instance, a child may inherit a factor for albinism but still appear normal physically. Why? Well, hold your horses, we shall see that shortly. So the concept of variation is a complex one, but interesting at the same time. At the very basic, variation refers to observable differences in living organisms. For instance, in garden pea, some have white flowers, others have red, and others have pink. In humans, variations include height, skin color, blood groups, and obviously sex, that is whether you are male or female. Variations can be continuous or discontinuous. Continuous variations exhibit a wide range of differences for the same characteristic from one extreme end to the other. For instance, in humans, if you consider a characteristic like height, in this group there is the shortest and the tallest. These are the two extreme ends, but in between them there are intermediates over a range. Other features that show continuous variations include skin color, weight, and fingerprints in humans. Height, number of leaves, and girth in plants also exhibit continuous variations. On the other hand, in discontinuous variation, there are no intermediates for a given characteristic. For instance, sex. You are either male or female, period. No one falls in between. Well, abnormalities do occur, but naturally it's not supposed to happen. The blood groups. You are either A, B, AB, or O. So in this variation, there are definite distinct groups of individuals with no intermediates. So exactly what causes these variations? Variations between children and their parents occur due to fertilization or when sperms and eggs fuse. Remember, sperms and eggs contain half the number of chromosomes. So after fertilization, the complete number of chromosomes is restored and the child gets half of its chromosomes from the mother and the other half from the father. So the child will bear some features that resemble the father and some the mother, and thus cannot look exactly like either. But it doesn't stop here. Sometimes the child may bear features that are in neither of the parents, or at least were not visible. The truth is, these features were carried by either of the parents in their genetic material but were not expressed in their physical or visible bodies. We shall see about phenotypes and genotypes later. Other than fertilization, variations also occur during gamete formation, either due to crossing over or independent assortment, or both during meiosis. Crossing over occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalence. Remember, homologous chromosomes share similar but not identical genes. Once this is formed, crossing over can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occur, leading to increased genetic diversity. Independent assortment occurs in metaphase 1. During metaphase 1, bivalents arrange along the metaphase plate. Due to independent assortment, this arrangement is random. What that means is that, for instance, here, having 3 pairs of homologous chromosomes, it is not certain that the reds go to the left and the greens to the right, or the first red to the left and the second to the right. No, the arrangement is random. So if a cell has 3 bivalents, the possible combinations are many, leading to genetic diversity. So imagine 23 pairs of chromosomes in humans. How many possible combinations? Inconceivable. Finally, other than during reproduction, variation can occur at any stage in life as a result of what we call mutations. And these are basically accidental changes in the DNA structure or composition. We shall look at mutations separately. The DNA. DNA is the genetic material that ultimately defines an organism. It is the hereditary material. DNA means deoxyribonucleic acid. The structure of DNA was phenomenally described and explained in 1956 by 2 scientists, Watson and Crick. This discovery is one of the greatest breakthroughs in the history of science that led to understanding of molecular biology. The DNA structure as we know it today is as was described by Watson and Crick. The DNA is a double-stranded polymer of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The backbone consists of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. It was also discovered that the amount of cytosine in a DNA molecule always equals to that of guanine, and that of adenine always equals to that of thymine. This conclusively led to what we call complementary base pairing. Cytosine always forms 3 hydrogen bonds with guanine, while adenine forms 2 hydrogen bonds with thymine. The DNA is then supercoiled to form a twisted ladder-like structure with the nitrogenous bases forming the stairs. The DNA stores the genetic information in a coded form. It allows the transfer of this genetic information unchanged to the daughter cells through replication. The genetic information is encoded or translated into proteins and defines the characteristic of living organisms. But how is the information coded? This is in the form of genes. A gene is a section of the DNA that codes for a certain feature or protein. For instance, the gene for hemoglobin, a certain enzyme, hair color, eye color that is brown or blue, sex, either male or female, height, and so on and so on. Each protein is coded by a unique gene. So you can imagine there are thousands of genes due to the thousands of proteins in human. In fact, there are over 100,000 genes in human. The part of the DNA constituting a gene is also called the coding region. However, a very huge section of the DNA does not constitute genes. This is called the non-coding region or junk DNA. If changes occur in the coding region or gene, there are consequences, but in the non-coding region, there are none. And this is perhaps nature's way of preventing fatal consequences of mutations. In fact, over 80% of the human DNA is junk. So with this numerous number of genes and junk sections of DNA, the molecule is of course very long and could even stretch several meters when straightened. So how then is it accommodated in a cell? The DNA does not exist as one long continuous strand. No. Rather, it is cut into sections which are then packaged together with some proteins to form the classic structures called chromosomes. For instance, the human DNA has been packaged into 23 pairs of chromosomes. Chromosomes are long thread-like structures found in the nucleus of a cell. Each chromosome is made up of two parallel strands called chromatids. Each pair of chromatids is connected at one point by a structure called centromere. In body cells, otherwise referred to as somatic cells, chromosomes occur in pairs. Each pair of chromosome has a characteristic length. The member of each pair is called a homologous chromosome. This means that the chromosomes are alike in appearance, although their genetic composition may be different. During sexual reproduction, each parent contributes one of the chromosomes of the homologous pair. Along the length of the chromosome is where we find this series of structures called genes. Genes are too small to be seen even with a powerful microscope. It is the genes that determine the characteristics of the cell and its progeny. All cells, including the gametes, that is sperm and ova, contain chromosomes. Counts of chromosome numbers show that there are a definite constant number of chromosomes in each cell for every species of animal or plant. For instance, in human, each somatic cell has 46 chromosomes, while each gamete contains 23. Upon fertilization, the number 46, called the diploid number, is restored in the zygote. In sheep, there are 56 chromosomes or 28 pairs. The cow has 30 pairs. Fruit fly or the Drosophila melanogaster has 4 pairs of chromosomes. Chromosomes are present in the nucleus at all times, but they only appear or become visible when viewed under a microscope during cell division, that is, during mitosis and meiosis. DNA replication. Growth in organisms occur due to increase in the size or number of cells. For cells to increase in number, division occurs in the process of mitosis. After cell division, each resulting daughter cell must contain the exact same genetic material as the parent. So before a cell divides, the genetic material is first duplicated, and this occurs during interphase. The ability of DNA to replicate itself clearly explains how inherited characteristics are passed on. The double helix structure of DNA is joined together through complementary base pairing between the two strands. Replication is accomplished by an enzyme called DNA polymerase. Each strand of the DNA molecule is used as a template for synthesis of another double strand. Replication begins with the DNA structure unwinding and the strands being separated by breaking the hydrogen bonds in the base pairs. And each parent strand is used as a template for synthesis of a new complementary strand by addition of nucleotides by the enzyme DNA polymerase. Due to the complementary base pairing, the new DNA molecule is exactly like the parent molecule. Replication begins at a specific area of the molecule called the origin of replication. It denotes the area of active replication called the replication fork, and several enzymes are involved. Replication simply involves the addition of nucleotides to a growing strand complementary to the parent one. DNA and protein synthesis. Basically, the DNA is responsible for protein synthesis in the cells. This bears a lot of significance since proteins control nearly every aspect of life in a cell. Proteins have both structural and functional roles. The hair and nails is a protein called keratin, and this is a representative of the structural proteins. Enzymes are all proteins in nature. Some hormones are proteins like insulin and growth hormones. Considering their importance in controlling and regulating cellular activities, these proteins must be continually synthesized by the cells to sustain life, and this is the role of the DNA. Proteins themselves are made of amino acids joined together to form a chain. Examples of amino acids include proline, lysine, phenylalanine, methionine, glycine, and many others. The DNA molecule codes for these amino acids with the sequence of bases along the DNA strand, acting as the alphabet or code that spells out the sequence of amino acids. The strands are decoded in triplets or 3 bases called codons. Each codon represents an amino acid. Protein synthesis takes place in the ribosomes, but since the DNA is contained in the nucleus and ribosomes in the cytoplasm, a means of communication must be established to instruct the ribosomes. When a protein is made, we say that the gene has been expressed. In expression, only the coding region is involved, unlike in replication. It involves 2 stages: transcription and translation. Transcription. During transcription, the coding region of the DNA is converted to a molecule called the messenger RNA, abbreviated mRNA. An RNA or ribonucleic acid is a single-stranded polynucleotide. mRNA is a complementary strand to the gene with one simple difference. It does not have the base thymine. Whenever thymine is to occur, it is replaced by another base called uracil. So imagine a gene section with the following sequence. The complementary strand would have the sequence that would make the DNA. However, using the same section to make a complementary RNA strand, remember that RNA is single-stranded. For mRNA, T is always replaced by U, so we shall have Once this mRNA is synthesized, it leaves the nucleus and into the cytoplasm to the ribosomes. And then we move to the next step, translation. Translation is the decoding stage when the mRNA is translated into an amino acid sequence by the ribosomes. So the mRNA sequence is read in codons, each representing a given amino acid. In the case of our strand, GCC is the first codon, and then GAA, GUG, and then the last codon is UCA. If we decode this, we shall have GCC codes for an amino acid called alanine, GAA codes for an amino acid called glutamic acid, GUG codes for an amino acid called valine, and UCA codes for an amino acid called serine. Translation begins by the ribosome binding to the mRNA. A molecule called transfer RNA transfers amino acids to the continuing chain of amino acid. The transfer RNA has a codon complementary to AUG codon on the mRNA and thus binds to it. Attached to the tRNA is the first amino acid, methionine. Another transfer RNA comes and is complementary to the second mRNA codon and bears the amino acid decoded by the codon. The methionine is then transferred to this amino acid. The first transfer RNA exits and the ribosome moves along the mRNA, and the next transfer RNA enters. The process continues and the amino acid chain elongates with the addition of new amino acids. This is elongation. The process will continue until a stop codon is encountered. This is usually UAG. A release factor enters and the elongation is terminated. The ribosomes dissociate and the protein is released. So notice that each gene has a start codon AUG and a stop codon UAG. The section between these two codons is called the open reading frame, ORF. Also note that different transfer RNAs transfer different amino acids. Heredity. Heredity is the transfer of genetic material from the parents to their offspring. Gregor Mendel, an Australian monk, is considered to be the father of genetics. Through his phenomenal work of experimentations, Mendel made incredible observations and discoveries that set the roadmap for further research in the field of genetics. Being a monk and of course having plenty of time, Mendel performed various breeding experiments and observed the variations in different characteristics of the garden pea. In the garden, Mendel observed that there were tall and dwarf varieties of the plants. So in one of his classic experiments, Mendel selected a group of dwarf plants and self-pollinated them by dusting mature pollen grains onto the stigma of the same plant. He then collected the resulting seeds and planted them, and he made a very interesting observation. Mendel noted that the seeds germinated and grew into dwarf plants only. He repeated the experiment for several generations and obtained the same results. What this showed is that dwarf garden pea plants could produce only their own type, and that is, they were pure breeds. He then proceeded and selected tall plants and self-pollinated them. He then planted the resulting seeds and observed that they grew into a mixture of taller and dwarf plants. He took seeds from the tall offsprings only and repeated the experiment for many generations until he obtained only tall plants. This way, what Mendel did is that he established a pure breed of tall plants. He didn't stop there. Mendel then cross-pollinated the pure breed tall garden pea plants and the pure dwarf variety. He planted the resulting seeds and observed that the offsprings were all tall plants. He crossed two of the tall offsprings and planted the resulting seeds. Again, Mendel made another interesting observation. He observed that the second generation consisted of a mixture of tall and dwarf plants. He counted those plants and noted that the ratio of the tall to dwarf plants was approximately or roughly 3:1 respectively. Mendel observed that the ratio was always obtained when a cross was made between non-pure breeds of tall plants. From his work, Mendel was able to postulate hypotheses that are now considered as laws governing inheritance. Mendel concluded that the characteristics of an organism are determined by hereditary factors which occur in pairs. Only one of a pair of such factors can be represented in a single gamete, and this is known as Mendel's first law, the law of segregation. Now, at his time, Mendel had no idea of genes, and so he called them factors. He postulated that these factors are located on chromosomes and they are transmitted from parents to offsprings via gametes. Adult plants contain a pair of factors controlling their size, but the gametes contain only one of these factors. Now, one would wonder how exactly Mendel was able to successfully carry out his experiments. You could arguably say that he was a monk and had plenty of time. But really, that's not it. In any case, there were many monks during his time anyway. First, Mendel used a very favorable material, the garden pea plant, which is normally self-fertilized, and this made it easy for him to employ cross-fertilization at will. The pea plant he used had several contrasting characteristics, that is, the color of the flowers, the height, and so on and so on. In his study, he focused on particular traits, while those before him had been attempting to determine wholesome heredity of each organism. And also, Mendel kept accurate data on all his experiments, and from the analysis of his data, he was able to formulate definite hypotheses. We move to the next item, that is monohybrid inheritance. Now, in his experiments, Mendel postulated that the inheritance of one characteristic like height is controlled by a single pair of hereditary factors contributed by both parents. And this type of inheritance is known as monohybrid inheritance. It involves the transmission of just one pair of contrasting characteristics or traits, such as tallness and dwarfness for height, or smooth and wrinkled for shape of seed coats. A single pair of hereditary factors known as genes controls such traits. Genes occur in pairs on chromosomes, and such gene pairs are known as alleles. Actually, alleles are gene pairs that code for the same but not identical characteristic. For example, the gene that codes for height, the alleles are one coding for tallness and the other codes for dwarfness. You realize that both code for height but different manifestations. The genetic constitution of an organism is known as its genotype, while the outward appearance of the organism is called the phenotype. The genotype of an organism is represented using paired letters or symbols, where the capital letter represents the dominant gene, while the small letter represents the recessive gene. Capital T represents the gene for tallness. During gamete formation or gametogenesis in dwarf plants, each gene in the pair segregates into different gametes. When the male and female gametes fuse during fertilization, the offspring produced contained the same number of genes as in each parent. The inheritance of dwarfness in the garden pea can be illustrated diagrammatically as a genetic cross, as illustrated next. So the parental phenotype, we have there the male is dwarf and the female is also dwarf. Parental genotype, we have the diploid there, TT for the female and also TT for the male. During gametogenesis, there is segregation and the gametes are haploid. So we have T, T, and also on the other side we have separate T's. Now in fertilization, the gametes fuse. So small t fuses with another small t to produce the diploid TT. Again the other t with another small t to produce another diploid TT. And it is the same for all the fusions. So we have all the dwarf phenotypes in the F1 generation of offsprings. Similarly, the pair of genes in the pure breed tall plants will segregate into different gametes during gametogenesis. Thus, when self-fertilized, the resulting seeds will have half the number of genes from each parent. So we have this genetic cross for the tall variety, the cross between the pure breeds of tall garden pea plant. Parental phenotype, the male is tall and the female is tall. That is how they appear. Parental genotype, the male is TT, T capital, and also the female is TT. It is purebred tall. During gametogenesis, there is segregation of chromosomes, and so they separate. And again, the cross that occurs during fertilization, there is the fusion of these gametes. The male gametes and the female gametes fuse. And if we repeat that, you realize again we have the F1 offsprings having TT capitals for all of them. So all of them are tall, at least in their phenotype, and also in their genotype. They are purebred tall. Now, when the purebred tall plant is crossed with the dwarf plants, the resulting seeds grow into tall plants only. These offsprings represent the first generation, known as the first filial generation, represented as F1. The dwarf characteristic is not represented in the offsprings. Let's see that cross. Parental genotype. The male is tall, purebred tall, and the female is purebred dwarf. So the dwarf is small t and the tall is capital T. Gametogenesis takes place and there is segregation. The gametes then fuse during fertilization. So capital T fusing with a small t to produce a diploid, T capital, T small, and it is the same case for all the others. So phenotypically, all the offsprings of the F1 generation will appear tall. In this genetic cross, the male plant is tall and the female plant is dwarf. If the cross is reversed, for the female to be tall and the male to be short, will attain the same results. However, when these F1 offsprings are self-pollinated, they produce offsprings that grow into a mixture of tall and dwarf plants. These offsprings are known as the second filial generation or F2 generation, and thus there are no intermediates between the tall and dwarf plants. So let's look at that genetic cross again. Now we take the F1 generation offsprings to be the parents in this cross. So parental phenotype, the male is tall, at least that is what it appears, and the female is also tall. Parental genotype, both the male and female have the tall capital T and small t in their genotype. During gametogenesis, these segregate into gametes. So the gametes, one is capital and the other is small, and it is the same case for the female. So the F2 generation, we perform the cross. Capital T fusing with a capital T produces a diploid, that is capital T for both. Again, a cross between the capital T and a small t produces a diploid with a capital and small. Again, a small t with a capital T. The small t from the male and capital from the female produces a diploid with capital and small t. And finally, we have the small t from the male fusing with the gamete small t from the female to produce a diploid with TT, small for both. Now, from these genetic crosses that we have looked, you notice that the following are components of a genetic cross. First, we have the parental phenotypes, and that is the outward appearance. Then we have the parental genotypes. The crossing of X should be shown here. The gametes, conventionally circled. The fusion process is the fertilization, and the failure generation genotypes are also indicated. A Punnett square can also be used instead of a cross and is shown on your screen. It is as simple as the cross one. Now, when the allelic genes are identical like capital T's or small t's, the condition is called homozygous. An individual with such a condition is referred to as homozygote. However, when the allelic genes are not identical, as in T capital and T small, the condition is referred to as heterozygous. An individual with such genotypic condition is referred to as a heterozygote. An individual with the genotype T capital T small will be phenotypically tall because the gene for capital T is dominant over the gene for small t. The allele small t is recessive. A dominant gene is that which can express itself in both its homozygous and heterozygous state, while a recessive gene is that which can only express itself in a homozygous state. Therefore, the genotypic condition capital T is referred to as homozygous dominant, while T small for both is homozygous recessive. The ratio 3 tall to 1 dwarf in the second generation is characteristic of monohybrid inheritance, where one gene is completely dominant over the other. This is complete dominance. The monohybrid crosses are based on Mendel's first law, which states that the characteristics of an organism are determined by internal factors which occur in pairs. Only one of a pair of such factors can be expressed in a single gamete. It is now known that Mendel's factors are specific portions of a chromosome called genes. It is also known that the process which produces gametes with only one of each pair of factors is meiosis. The process of segregation is accomplished during meiosis. Now, similar monohybrid results as those of Mendel have been obtained by using the common insect, the fruit fly or Drosophila melanogaster. The insect has many observable characteristics that show discontinuous variations, and these include the wing length. There is the long wing which is dominant over the vestigial wing. Eye color. Red eyes are dominant over white eyes. Size of the abdomen. The broad abdomen is dominant over the narrow abdomen. Body color. Gray body color is dominant over black body color. The fruit fly is a suitable organism for genetic study due to the following reasons. First, the female lays many eggs, resulting in a large number of offspring. This increases the sample size for study. The larger the sample, the more reliable the results. It also has many observable characteristics that are distinct and contrasting. It is also easily bred in the laboratory with minimum requirements. It has a short generation time, that is 10 to 14 days, and therefore many generations can be studied in a short period. The offsprings can be crossed with their parents at will, and this is called back crossing. The flies are safe to handle because they do not transmit any known human disease. Next, away from monohybrid inheritance and complete dominance, we now look at incomplete dominance. Now, in the experiments discussed so far, one allele has always been dominant over the other. For instance, in the garden pea, the tall plant with genotype capital T homozygous could not be distinguished from an individual with genotype T capital T small that is heterozygous. Thus, although the genotypes were different, the phenotypes were identical, indicating that the gene determining tallness is completely dominant over that determining dwarfness. However, in some plants such as the 4 o'clock plant, a cross between red and white flowered plants results in the F1 generation, with all the offsprings bearing pink flowers, thus showing blending of color. Self-pollinating the F1 individuals gives phenotypic ratio of 1 red, 2 pink, and 1 white. This shows that there is no allele which completely dominates the other, that is, both alleles express themselves equally. This is an example of incomplete dominance or codominance. Incomplete dominance can also be observed in animals. For example, in a certain species of domestic birds, a cross between a black-colored or black-feathered chicken with a white-feathered one produces offsprings with both white and black feathers. In genetic crosses involving incomplete dominance, 2 different capital letters are used to represent the 2 genes. So let R represent the gene for red flower color and white be represented by W. So parental phenotype, the male is red flowered and the female is white flowered. Remember, it doesn't make a difference which becomes the male or which becomes the female. Parental genotype, RR capital for both and white. Gametes, the segregation there, we have the gamete R, 2 of them, and then on the female side we have white. The fertilization fusion of both will produce RW for all the 4 offsprings of the F1 generation. Now, the RW genotype of the F1 generation represents neither the red nor white, but pink. Now, if we self the offsprings of the F1 generation, we have parental phenotype. Both are pink. So we have the parental genotype RW for both the male and female. Gametogenesis takes place and there is segregation, so we have gametes R and W for both of them. Then during fertilization there is the fusion, so R fuses with R in the first fusion, so we have RR in the F2 generation. Then R fuses with a W to produce RW. Again, W fuses with R to produce another RW, and then finally W fuses with another W to form WW. So we have RR is red flowers, 2RW are the pink flowers, and WW 1 white flower. So the ratio of red to pink to white is 1 to 2 to 1 respectively. Inheritance of the ABO blood groups. Now, in humans, the ABO blood group system is an inheritable characteristic that is transmitted from parents to offsprings. Unlike other genetic traits determined by a pair of alleles, the ABO blood groups have 3 genes involved, a case of multiple alleles, and they are responsible for the presence of antigen types on the red blood cells. These are genes A, responsible for the presence of antigen A, gene B, responsible for presence of antigen B, and gene O, responsible for 0 or no antigen on the red blood cell. Genes A and B have equal degree of dominance, that is, they are codominant and will both express themselves when present together in the AB blood group. Gene O is recessive and will only express itself in the homozygous condition. The genotypes for the 4 blood groups in the ABO system are thus formed by allelic pairs of genes as shown on your screen. These alleles are transmitted from parents to offspring in the usual Mendelian fashion. Thus, a marriage between a man of blood group A and a woman of blood group B will produce children of all 4 blood groups as shown below if both parents are heterozygous. Look at this cross on your screen. Another important aspect is the inheritance of the rhesus factor. Now, in human beings, the possession of rhesus antigen makes one rhesus positive, while the absence makes one rhesus negative. The presence of the rhesus antigen is due to a dominant gene which is dominant over the gene for its absence. If one of the parents is rhesus positive, then all the children will be rhesus positive, because the rhesus positive gene will dominate the rhesus negative. So rhesus negative children will only result if both parents are rhesus negative. Next, we look at determining unknown genotypes. Now, in the garden pea we saw, the gene that determines red flowers is dominant over that which determines white flowers. A plant with red flowers may either be homozygous or heterozygous for this characteristic. Now, to establish the correct genotype, it is crossed with the homozygous recessive plant. In such a cross, if all their offspring bear red flowers, then this indicates that the red flowered plant is homozygous, that is, it is from a pure line. Now look at this cross. All the offspring of the F1 generation have red flowers. This could only be so if the unknown genotype is homozygous for the dominant gene for red flowers, so that when it is bred with the heterozygote for the recessive gene of white flowers, all the offspring will be heterozygous, but the dominant gene will be expressed. If the unknown plant was heterozygous, the story would be different, as illustrated on this cross. A cross between an individual of unknown genotype with an individual of recessive genotype is known as a test cross. A test cross where an offspring is crossed with one of its parents is known as a back cross. Unknown genotypes can also be determined by carrying out selfing experiments. For example, a phenotypically tall plant is either genotypically homozygous or heterozygous for this trait. If selfed and all its offspring are phenotypically tall, then the parental genotype is homozygous tall. However, if after selfing both tall and dwarf offspring are produced in the ratio of 3 to 1, then the parental genotype is heterozygous tall. Sex determination. Now, sex determination in higher animals is controlled by a specific pair of chromosomes. For instance, in humans, there are 46 chromosomes, that is 23 pairs of homologous chromosomes in every body cell. The genes that determine whether a child becomes male or female are located on specific pair of chromosomes called the sex chromosomes, which are of 2 types, X and Y chromosomes, named after their shapes. The remaining 22 pairs of chromosomes are called autosomes and are responsible for other inheritable characteristics. A male human being carries the XY heterogametic, while a female carries the XX or homogametic combination. After meiosis in a male, the spermatozoa can either contain the X or Y chromosome, while the female ova will contain only the X chromosome. The sex of a child is a matter of chance and depends only on whether the spermatozoon that fertilizes the ovum carries the X or Y chromosome. So clearly, it is the man who determines the sex of the child. During fertilization, there are 4 possible genotypic combinations. So parental phenotype, of course we have the male and female. Parental genotype, for the male it's XY and the female is XX. The gametes fuse during fertilization and we have XX. XX, those are 2 girls. Then we have XY and XY, 2 boys. So phenotypic ratio of girls to boys is 2:2, which is 1:1. In terms of probability, the chance that a boy or a girl is produced in a family is a half. This means that chances are equally likely that a boy or a girl is born in a family. In other animals, the male is homogametic while the female is heterogametic, like in birds. Boy birds are XX while the girl birds are XY. In some insects, the female is XX and the male is X0. The Y chromosome is missing altogether. In the fruit fly, that is Drosophila melanogaster, sex determination is exactly as in human beings. That is, males are XY and females XX. Linkage. Now, an organism has a large number of characteristics controlled by many different genes, and due to the limited number of chromosomes, each gene cannot be located on its own chromosome. Therefore, chromosomes must accommodate many different genes, each controlling particular characteristic. For example, genes located on only 23 pairs of chromosomes control all the thousands of characteristics in human beings. Those genes found on the same chromosome are said to be linked. They are called linked genes. All the linked genes constitute a linkage group. Linked genes are inherited together and therefore do not segregate during meiosis. They are therefore transmitted in the same gamete. Cases of linked genes have been studied in Drosophila species. It has been found that the genes for wing length, abdomen size, and body color are located on the same chromosome, and therefore these characteristics are usually inherited together. Sex-linked genes. All genes located on the sex chromosome are said to be sex-linked, and therefore are transmitted together with those determining sex. In humans, there are few genes located on the Y chromosome which control characteristics that are exclusively male. These are premature baldness, tufts of hair in the ear pinna and in the nose. Characteristics controlled by genes located on X chromosomes include color blindness and hemophilia. These characteristics can arise either in males or in females. So it is easy to understand why color blindness and hemophilia are common in males. And this is why. Because the gene occurs on the X chromosome and males have X chromosome, there are no carriers in males. Once the gene for color blindness occurs, the male is phenotypically colorblind. However, in females with 2 X chromosomes, one can be a carrier if the gene occurs on one of the X chromosomes. Normal vision is dominant over color blindness. So, if N capital represents the gene for normal color vision and N small represents the gene for color blindness, now we can have the following cross. This is a marriage between a man who is colorblind. Notice that the gene is on the X chromosome. There's none on the Y chromosome. This man marries a woman who is normal. She has normal color vision, neither carrier nor colorblind. A cross between them will reveal that this couple will get 2 daughters who are carriers. Of course, they will appear normal, they will have normal color vision, and then they will of course have normal sons. Now look at the other case here. In this case, we have a man who has normal color vision and then marries a woman with normal color vision, but then is a carrier of the gene for color blindness. So a cross between these two, let's see what kind of children these couples get. Their offsprings there, we have a daughter with normal color vision, not a carrier. And then we also have another daughter who is a carrier. There is the gene for color blindness on one of the X chromosomes. And then we have a son who has normal color vision. The gene on the X chromosome N is capital for normal color vision. But then they will also get a son who carries the gene for color blindness on the X chromosome. And that son automatically becomes colorblind. Now, important on the issue of color blindness or sex-linked genes, it is important to note where to place the gene. Some genes are only located on the X chromosome and others only on the Y chromosome. Of course, the genes located on the Y chromosomes are very few and often not very vital for survival of the organisms. Now, in your own time, perform a cross for the condition of hemophilia. Remember also, the gene for hemophilia condition is also located on the X chromosome. So you can attempt to perform several crosses with either carriers or homophiliacs for either both or males or females. Remember, there are no male carriers for color blindness and hemophilia. A male is either normal or colorblind or hemophilic. So that next we look at mutations, which we call accidental changes in the structure or composition of the DNA. Mutations or errors that occur in the replication of the DNA can have significant impacts on individuals or even the entire species. Mutations that occur within the body or somatic cells can, for example, cause illness such as cancer, if the mutations program the cell and all its daughter cells to grow and divide uncontrollably. Such mutations are often caused in humans by radiations and chemicals such as those found in cigarette smoke that break strands of DNA. As we shall see, the corrective repair work often leaves the DNA with an error. Mutations that occur in sex cells do not affect the individual in which they occur, but rather, if they are carried in a sex cell that unites with another during fertilization, they are carried into the next generation. This is because the individual who inherits the mutation has it in every cell of your body, including those that will be eventually used in the production of sex cells. If a mutation occurs in a somatic cell, the mutation is not inherited by the individual's offsprings. For example, if a man develops skin or lung cancer later in life, his children born after will not inherit the cancer. This is because the abnormality is only in the skin and lung cells, but not in the sperm cells. Not all mutations are harmful to individual organisms. Continuous use of insecticides or antibiotics could cause mutations in the insects or bacteria. They are no longer affected by the chemical. Whereas this is bad news for the humans, it is favorable to the bacteria or insect targeted by the chemical. Resistance in insects would take long to develop because they have many cells, and for a mutation to be inherited, it has to affect their reproductive cells. In bacteria, resistance is rampant because they are single-celled. A mutation in a parent cell is inherited by the daughter cells after cell division. Resistance to antibiotics, like the case of tuberculosis, is a major concern in healthcare provision. However, some mutations may render the organism unable to perform physiological processes well, or in some cases completely, often leading to fatal conditions and even death. Still, some mutations may have absolutely no effect on the organism, like those that occur on the junk or non-coding region of the DNA. This last case is more common than the other two. There are three basic types of mutations: point mutation, insertion mutations, and deletions. Occasionally, during replication, in spite of all the proofreading and correction, a mismatch in base pairs in one of the chromosomes can occur. See here, a red has been paired with a green, which is obviously an error. If this goes unnoticed, the cell proceeds with the entire mitosis or meiosis. Repair enzymes in the daughter cells that receive the mismatched pairs notice the problem and proceed to correct it. However, since this is taking place in the daughter cells, these enzymes have no way of knowing which strand was the original parent strand and which is the daughter strand. The enzymes have two options. Either it replaces the green on the lower strand with the blue, which will match the red base, or replace the base on the upper strand with a yellow, which will also produce a match. Sounds easy, but it brings a difference. If they correct the mismatch by replacing the base on the daughter strand, the original base pair will be restored. However, if the enzymes correct the mismatch by replacing the base on the original strand, a different DNA structure from the original will result and a substitution mutation will have occurred. Insertions and deletions occur not due to any error in replication, but rather from a break or breaks in one of the strands. Often outside sources such as radiations and chemicals cause these breaks. Insertions often occur when one strand breaks in a single location and one or both of the free ends loop out. Repair enzymes fill up the gap with new nucleotides, making this strand longer than the other. When replication takes place and the cell divides, one daughter cell receives a longer DNA and the other receives the normal unchanged DNA. Deletion mutation occurs if a strand of DNA breaks in two places. The section on the break is forced out and the repair enzymes pull the remaining ends together without filling in the missing nucleotide. Again, a loop occurs in one of the strands, but in this case in the unchanged strand. Now when cell division occurs, one daughter cell receives a shorter segment of the DNA due to the deletion mutation, while the other remains unchanged. Mutations are very fatal when they occur in a gene section, because when the gene is translated into a protein, it may result into a completely new protein due to the changes in the codon. If you look at this DNA ladder, and then we have the mRNA strand after transcription, and then the codons are translated into amino acids. So if I substitute this guanine for, let's say, adenine, when I transcribe that into mRNA, that becomes a U. So what does that mean for the amino acid? The original codon CCU codes for proline. If you look at the codon chart, that is CCU proline. But if we change that code, we change G to A, which changes C to a U, then the codon becomes UCU, no longer codes for proline. In fact, it codes for serine. UCU serine. So that single point mutation changes the amino acid sequence we have. Now look at another base substitution. What if instead of this A we have a G? that changes this codon from CCU to CCC. And CCU translates to proline. What about CCC? If you look at the codon chart, we say that CCU is proline, but CCC is proline as well. So sometimes a substitution has zero effects because of the redundancy of the genetic code. So that's what base substitution looks like. A classic example of a condition caused by point mutation is sickle cell anemia. This is a section of the DNA coding for the protein hemoglobin, a normal hemoglobin. It is made up of 154 amino acids. Sickle cell anemia is caused by one wrong amino acid. The effect is magical. It causes complete distorting of the normal hemoglobin. Remember, hemoglobin is the carrier molecule that transports oxygen in the body. When the DNA strand is transcribed, we will make this mRNA, which is now ready for transcription. Then, the codons are translated into amino acid chain: valine, histidine, leucine, threonine, proline, and 2 glutamates at the end. Now, this is a normal hemoglobin. And if your hemoglobin is normal, your red blood cells are nicely biconcave and very efficient with a normal lifespan of 120 days. Now, in sickle cell disease, there is a substitution or a misspelling. In a normal cell, the codon is CTT, but here I have CAT. Now, this is transcribed into mRNA and that codon becomes GUA instead of GAA. Now, in the translation, GUA codes for valine and not glutamic acid. So, in the sickle cell anemia, glutamic acid is replaced by valine. And this brings a lot of trouble. It produces an abnormal hemoglobin, and the red blood cells assume a crescent or sickle shape with a short lifespan, and this is a life-threatening condition. Perfect. Let's now see base insertion and base deletion. We'll start with base insertion. In fact, we will just do base insertion because it kind of have the same effect. So if I were to add a T into this code, let's push that T in there. Now look at how it changes things. When we read the DNA code, we have to read it in triplets, but I have 4 letters here, so I have to shift the reading frames. When we transcribed, that will also shift our reading frame on the mRNA as well. And when we translate, that's gonna change many of our amino acids, and it's gonna mess up because it even made a stop codon in the middle here. But the point is that it leads to what we call a frame shift. So a simple addition or insertion of a base leads to a whole new frame. And if we delete a base again, the frame will shift in a different direction. And the resulting protein is a nonsense one or a non-functional protein. We call it a nonsense protein. Now let's move to another type of mutation, chromosomal mutation, and specifically changes in chromosome structure. And here we have deletion, duplication, inversion, and translocation. We start with chromosome deletion. It happens when a section of chromosome is missing. So if we have this structure of chromosome with the letters representing the many genes, for deletion we have lost a section of the chromosome. In chromosomal duplication, it is when a section of the chromosome is repeated. So maybe this happens. You can see the repeated section here, the B and C. The B and C are being repeated or duplicated. In chromosomal inversion, a section of a chromosome is inverted. So it breaks off, turns over, and reattaches. Now, we didn't lose any gene and we didn't gain any either, but we changed their order. Instead of A, B, C, D, we have C, B, A, D, and in most cases, order is important. Lastly, in chromosomal translocation, a section of a chromosome breaks off and attaches to another non-homologous chromosome. So this other chromosome has a whole new set of genes on it, and in translocation, a part of the chromosome breaks off and relocates or translocates to a non-homolog. And again, we didn't lose any genes, but we changed our linkage pattern. Another type or class of chromosomal mutation is change in chromosome number, and we look at non-disjunction. Now look at this karyotype for a moment and look clearly. Do you realize anything unusual in this picture? Anything out of the ordinary? Look now. We call this trisomy 21. We are supposed to have 2 chromosomes of each because we get one from either of our parent. But for some reason, we have 3 on chromosome 21. This is called Down syndrome. Having 3 chromosomes is a pretty nasty genetic disorder, but let's see how it happens. But before, just hold on. Do you see anything unusual in this karyotype? Pause the disc if you need to. How about you look down here? We have an extra sex chromosome. We got 2 X's and 1 Y. This is called Klinefelter's syndrome. Here I give one more. See if you can point out the error. And I give you another little hint. We are missing a sex chromosome. We have an X and nothing. This is called Turner syndrome. What all these have in common is that by a non-disjunction, we have an improper chromosome number. So let's see how this might happen. If you look back at meiosis here, we have a germ cell that's going to give rise to a gamete. We know we are going to replicate our DNA. We get to prophase 1 of meiosis. Tetrads form and homologs pair up. In metaphase 1, they align at the center, and in anaphase 1, homologous chromosomes separate. Of course, except when they don't. This is disjunctions. The chromosomes have separated. Non-disjunction will be a failure of these chromosomes to separate. So what if this happens? See the mistake? This is non-disjunction. When homologous chromosomes fail to separate at anaphase 1. We finish meiosis and see what we shall have. We go to telophase and then cytokinesis and into meiosis 2. And notice these duplicated chromosomes should be over here. But the cell continues on with division and we move forward. And at the end, we have our last cytokinesis and we have our gametes, which naturally are supposed to be haploid. But these 2 are missing 1, and these other 2 have an extra chromosome each. So non-disjunction results in the formation of gametes with the wrong chromosome number. We can summarize the process here. So we see the non-disjunction process here during meiosis 1. So human genetic conditions caused by non-disjunction include Down syndrome or trisomy 21, XXY or the Klinefelter syndrome. We also saw the Turner syndrome where we had the X chromosome with no partner. And we can also get 3 X chromosomes, and we call this metafemel syndrome. And we have the XYY condition. So notice how these have to do with the X and Y chromosomes. But why is there no monosomy 21 syndrome? Because really, whenever we have a trisomy, we should have a monosomy event. And the answer is that having an extra chromosome seems to be a viable disorder, and that a cell can still survive. But missing a chromosome is not a viable or tolerable disorder, and often leads to death of the cell. Why is there no Y-naught syndrome? We have X-naught syndrome. The answer is that there are too many vital genes on the X chromosome but very few on the Y chromosome. Thus, if the X chromosome is missing, the cell cannot survive. Why is there no trisomy 14 syndrome? And I just picked the number 14 randomly. But we know of trisomy 21. It is a universally known disorder, but we have no documented evidence of trisomy 14 or any other for that matter. It doesn't happen, or maybe only trisomy 21 is a viable offspring. Two more questions before we wrap up. Can you survive without the Y chromosome? Pause the disc and think about it. I know you already answered that. Yes, you can. Women do it every day. There are no crucial genes on the Y chromosome. And the last question, can you survive with only one X chromosome? Well, yes, of course. Men do it every day. It makes sense. All these conditions are viable. These are genetic defects. There are lots of people walking around with these conditions and you never notice it. They don't convey too many outward signs and certainly no disabilities. Take time to look at other genetic disorders caused by mutations like sickle cell anemia, hemophilia, and albinism. I hope you learned something.
Without many of these discoveries and inventions, few of us would be alive today. Welcome to WatchMojo.com, and today we're counting down our picks for the top 10 greatest medical breakthroughs in history. Number 5. DNA. Though DNA was isolated in the 19th century, its helix structure was only accurately identified in the middle of the next century. These cellular molecules are now known to carry our genetic code, which allows all living organisms to exist, develop, and procreate. The discovery of deoxyribonucleic acid has since led to the development of gene therapy and opened doors in fields such as forensic science, biotechnology, and more.